So ConeSight is a startup company. We're preclinical still, from the lab of Michael Young at Harvard Medical School. And we're really excited about our cone progenitor cell therapy because it's time. The eye is a very active space for gene and cell therapy, but to date, cell therapies have been largely limited to retinal pigment epithelial cells, which are the support structural cells for cones and rods, cone photoreceptors. Part of the reasons why cones have not been attempted in clinical trial is that until now they've been really hard to regenerate from stem cell products, whereas retinal pigment epithelial cells are very easy. But it's our contention that for the inherited retinal diseases that start primarily in cones, as well as the ones that start in rods and progress to cones, that unless you replace the cones themselves, you're not going to make significant headway in the disease. So the RPE can slow cone regeneration in theory, but never replace the cones. So our clinical approach is to start with cone rod dystrophies as our first clinical target because the disease and the cell product are so perfectly matched. However, we are planning rapid expansion into the rod cone dystrophies, which are more common, like retinitis pigmentosa, because the same preclinical package can be adapted to the rod cone dystrophies. And ultimately, we think these cells will be extremely useful in treating macular degeneration, but in combination with RPE. So discussions are ongoing about how to combine these technologies and target the really huge unmet need for macular degeneration.